Good afternoon, everyone. Thank you so much for the invitation and for having me here today. I'm so glad to learn more about all these fascinating technologies. So in my lab, we do subcellular imaging. And today I'm going to tell you about a project that try to understand how a particular type of organelles, endosomes, are able to sort membrane proteins for degradation. And so let me give you some context of what we are talking about. So pretty much all cells need to be able to regulate the composition of the plasma membrane as an interface that allows them to interact with the environment, with other cells, with the tissues and organs. And so in this diagram, I'm representing here the plasma membrane, the surface of the cell. And these green noodles here represent plasma membrane proteins, receptors, ion channels, enzymes. So eventually when they need to be degraded because they are not functional any longer, or because maybe these are activated receptors whose signals have to be downregulated, these proteins become ubiquitinated. They are internalized by endocytosis, so they are sorted into vesicles at the plasma membrane, sent inside the cell, and eventually they will reach an organelle called multivesicular endosomes. So first they are located in the limiting membrane on the surface of these endosomes, but quite quickly they are recognized by a group of protein called escorts, endosomal sorting complex required for transport, that can bind to those ubiquitin moieties. They can cluster all the proteins in one given spot and sequester them into intraluminal vesicles. So these vesicles are now inside the endosome. And therefore, when the endosome fuses with the vacuole, all these vesicles can be released and properly degraded. And that's how plasma membrane proteins are degraded in all eukaryotic cells. It's kind of a complicated process that requires more than 60 proteins in total, not only the escort proteins. But when this process fails because the escort proteins are not functional, then the plasma membrane proteins remain on the surface of the endosome, on the limiting membrane. And when this organelle fuses with the vacuole, the plasma membranes are now on the surface of the vacuole. They cannot be degraded. And again, if this is a receptor telling the cell to divide, it's easy to understand why mutations in escort proteins are connected to cancer, for example, in humans. So the organelles are quite small, as you can see in this TEM image. They are around 250 nanometers in plants, and the vesicles inside are around 40 nanometers. So we need to use transmission electron microscopy to see them. We're quite interested in this process because it's quite different from other type of vesiculation events like endocytosis, where these vesicles are pushing and forming towards the cytoplasm. These vesicles are forming away from the cytoplasm. They are being bent away from the cytoplasm. And this type of vesiculation event in nature has been shown to be only mediated by escort proteins. Nature has come up with only one solution for this type of events, and is conserved all the way from archaea to eukarya. And so just zooming in to see a little bit more detail what that entails, these cartoons represent the surface of the endosomes, and these orange lollipops here are plasma membrane proteins that contain their ubiquitin. And so if this is a cytoplasmic side and this is a lumen of the endosomes, the escorts are recruited from the cytosol. They are all soluble proteins. They are recruited sequentially, escort 0, escort 1. They bind the ubiquitin on the plasma membrane proteins, escort 2. And we start to see at this point the clustering of plasma membrane proteins, cargo proteins. And finally, when escort 3 proteins are recruited, they tend to polymerize in long spirals that help to bend the membrane. We have done cryo-electron microscopy imaging of some of these escort 3 proteins. They form very tight spirals that can build quite a bit of potential energy. And so as they are polymerizing on the surface of the endosome, they finally cause the buckling of the membrane. So the first vesicle bud formed towards the interior of the endosome. And then the ubiquitin is removed from the cargo protein, from the plasma membrane proteins. And the last complex that is recruited is a chaperone called SKD1, and is positive regulator LIP5. So this complex now is able to remodel these SCOR3 filaments to further constrict the neck to the point that the vesicle can be released. This model that is based on these studies propose that one of the functions of this complex SKD1-LIP5 is certainly to constrict the neck, but also to recycle all the escorts back to the cytosol. And so the escorts are fully recycled, and the vesicle is free in the endosome through that same process. Why this is important, why we care about? Well, essentially, it's so important that no eukaryotic cell can fully survive without being able to complete this process successfully. And in plants, we have different types of mutants. Some mutants that are affected in some peripheral or accessory escort proteins have mild growth defects, but some others are very extreme. They are either embryo lethal or seedling lethal. And as you can see, there are issues with the differentiation of veins, organs. This is a root meristem in an escort mutant, completely disorganized. Cell differentiation is affected. And for those that are able to form flowers, they are usually infertile because the pollen grains are not able to survive. So it is certainly an important process, and it still is quite unclear how it happens. Again, because of this opposite topology and the challenges of understanding how the constriction happens and what is the function of all these individual proteins. So what we are using is pretty much, this is our imaging pipeline. We use high pressure freezing as a way to preserve tissues with in a much better way, with a much higher fidelity. And so we do high pressure freezing, freeze substitution, resin embedding, sectioning, and then we do three-dimensional imaging through electron tomography. And finally, we calculate those tomograms, segment, and analyze our endosomes. So the first thing that I'm going to show you is a wild-type endosome from Arabidopsis, from an Arabidopsis root cell. And we are going to move through the tomogram to see the organelle. So this is a limiting membrane that I mentioned before, and you can see all the vesicles inside. And one thing that we notice analyzing these tomograms is that actually the vesicles, some of them are connected to the limiting membrane, but most certainly, and now it's going to play back, all these vesicles are connected with each other as well. And you can see the connection through all these vesicles. They are forming very complex networks. And as we segment these tomograms, this is the images that we find. So the vesicles form in specific domains on the surface of the endosomes, and they are not fully separated ever. They form in networks. There are constricting necks between these vesicles. And again, we don't see any evidence of vesicle separation. And as this process is essential for life, we were very interested in understanding what is going on. Is there something peculiar about plants, or what may be the function of this concatenation, this system of budding that establish necks and bridges between vesicles? So we detected two main patterns. They can either form linear change of vesicles by keeping the same budding site. So there is one vesicle form, one neck, another one form on top, and so on. Or there are instances when we see multiple budding sites. So here there are, for example, in this electron tomographic slice, you can see two vesicle buds that are connected by a bridge. If we see the three-dimensional reconstruction, we can see that actually there were three. And so these are quite common in Arabidopsis as well. So two types of budding leading to vesicle concatenation in endosomes. So we can use these endosomes to then apply models and understand how the dynamic of vesicle diffusion, protein diffusion could happen in these organelles. Basically, what we were asking here is that we have a very different situation from the model in yeast. We see single buds, double buds, 3 buds in linear chains. Would it be possible to recycle the escort? That is part of the dogma on how escorts work. If I remove escort from this neck or from this neck, would it be able to still keep the cargo inside? So we use these geometries to run a simulation. And so we know that one of the plasma membrane proteins that are sequestered into these vesicles for degradations are PIM proteins. We heard from Tim earlier during the sessions that PIM proteins are important for auxin fluxes and for the formation of floral buds. Well, these proteins are also degraded through the endosomal pathway. And so we calculated the diffusion rate. And using these tomograms, we were able to run simulations on how long it would take for those PIM proteins to escape from the 3 geometries if the escort were not there and nothing was stopping them from escaping these structures. And what we found in all cases is that it doesn't matter how many concatenated vesicles you have, they would be escaping half of them at least in less than 100 milliseconds. So that means that the vesicles by themselves don't have enough capacity to retain the plasma membrane proteins. And we predicted that the escort proteins need to stay in the necks. They cannot be recycled, otherwise the endosome wouldn't work. And we were able to prove later on that the escort proteins indeed are retained inside those necks by immunoglobulin labeling and some other biochemical assays, and that their presence is very important for keeping the cargo inside those vesicles. So quite different from what we see in yeast, where vesicles form one by one, and the escort proteins are recycled. Plants don't recycle their escorts, and they form networks of concatenated vesicles. So you may be thinking, well, how is that important for the function of these endosomes? And so to answer that question, we have been looking at escort mutants in Arabidopsis. And so here there are 2 escort mutants. Chin1 is one of the escort 3 filaments, and LIP5 is the positive regulator of the chaperone that is needed to constrict the necks. And the 2 plants you can see here, LIP5 is a mild a mutant. It has some growth effects, but overall it's a fertile normal plant. Chin1 is the one that dies as an embryo or a seedling. At the subcellular level, we can express these PIM proteins fused to a green fluorescent protein and see their localization. You can see in wild type samples, the PIM proteins are in the plasma membrane as expected, both for references for Chin1 and LIP5, but in the mutants, they are relocated to the vascular membrane. As I showed you before in our diagram, that if the escorts fail to sequester them into vesicles, later on they are going to end up in the vascular membrane and fail to be degraded. So both mutants are failing to sort plasma membrane proteins into vesicles. But how these endosomes look like? So we did electron tomography. These are all wild-type tomograms. We just placed some red spheres inside the vesicle, so it's easier to visualize. And you can see large clusters of concatenated vesicles. In the mutants, both LIP5 and Chin1, we see both a reduction in the number of vesicles, but also they are not concatenated, they are free. And so we can quantify these features in our tomograms. And what we are seeing, for example, is that there is a reduction in the proportion of vesicles that are concatenated. So this diagram is showing us in blue concatenated vesicles, free vesicles in red, and single bud like this in green. So you can see that around 80% of the vesicles in wild type are concatenated, but those numbers are drastically changed in the 2 escort mutants with a predominance of free vesicles. So concatenation is lost, and that correlates with the lack of capacity to retain cargo inside those vesicles. Not only that, the other feature that we notice is that these mutants are lacking or have a drastic reduction in multiple budding sites. I showed you before that in some cases, we see the formation of these multiple budding sites where multiple buds form simultaneously, and they are interconnected with each other at the limiting membrane. And they can be 2 vesicles involved or 3 vesicles engaged in a multiple budding site. We can do these quantifications in the wild type. You can see that the proportion of vesicles engaged in different type of buds, up to 6 vesicles can be all together in one single budding site. But those multiple budding sites are completely lost in Ching1 and almost gone in LIP5. So both the loss of multiple budding sites and concatenation correlate with the lack of capacity to retain plasma membrane cargo proteins like the PIN proteins in these endosomes. So we were interested in understanding how these features evolve within plants. So yeast seem to be sorting their plasma membrane proteins in a very different way. So why is unique in plants, and when this happen during evolution? And so we have been sampling pretty much across the phylogenetic tree of plants. So we have tomograms of multivesicular endosome from yeast, from Cara within algae, Marcantia, and you name it. So all the species that you see, or genus that you see with genera that you see with asterisks, we have samples. And so we have reconstructed all these tomograms and just asked some questions about what mechanism of vesiculation they use to sort the plasma membrane proteins. So when we look at yeast, what we can see is that, as it has been reported before, around 86% of their vesicles are free. There are no multiple budding sites. There are only a few concatenated vesicles. And again, that's the traditional model. Cara is not different from yeast. 83% of the vesicles are free. Now, when we get into land plants, Marcantia, we see a change. Now, 26% of those vesicles are concatenated, although we don't see multiple budding sites. For ferns and conifers, we see an increase in the proportion of concatenated vesicles, 40% for terrace, 52% for pine. And now in pine, we see the first few examples of multiple budding sites in a very low proportion. When we get into Arabidopsis, now most of the buds are involved in some kind of multiple budding sites. Single budding sites are a minority, and 84% of the vesicles are connected. So during evolution, there were several changes. Concatenation became predominant, and between conifers and flowering plants, somehow they changed the way that they were vesiculating their endosomes, and we see this predominance of multiple budding sites. So we went in more detail and tried to see exactly when that change happened. And so we were sampling here among the most basal flowering plants, Amborella and water lilies. And so if we look at the endosomes of Amborella, they look very similar to pine. Very low proportion of multiple budding sites and 53% of concatenated vesicles. When we get into water lilies, now they look identical to Arabidopsis. So something happened in this transition that changed drastically how these endosomes work. And what we are trying to see now is exactly what that was. And we are resourcing against to modeling. So in collaboration with Angela Sarik from the Institute of Science and Technology in Austria, we have been performing some simulations to understand exactly what is different between these vesiculation processes. So Angela developed a model for yeast mostly, where they can simulate, and what we have here in green is the limiting membrane of the endosome. These are the escort-3 spirals. They can change the affinity, the binding affinity between the escort filaments and the membrane. They can change the curvature of how that filaments interact with the vesicle and the curvature of the filament itself. And then in the way that they simulate the presence of the plasma membrane protein is by placing these big spheres on the surface. They are just giving the model the information where the plasma membrane proteins are and changing the properties of the membrane locally. So we asked Angela, what would you have to change in the model to go from single budding to concatenated budding? And pretty much the answer was not much. If what you want to do is a linear change of vesicles. So if there are enough cargo proteins, enough plasma membrane proteins, and enough escort proteins on the surface, this will happen spontaneously. You will end up having concatenated vesicles in linear change. Now, for the multiple budding sites, things change quite dramatically. And again, to remind you, this happens somewhere between Amborella and the rest of the flowering plants. So they have been running these simulations and trying to understand how we can build it like a double budding site. And it needs to break the spirals, at least in multiple pieces, and a pore has to emerge between the two vesicles. And for that to happen, there has to be a drastic change in the binding energy of the escort to the membranes and the curvature of the escort filaments. But all these properties are connected to escort-3 proteins. So what we are doing now is carefully going through all the sequences of the escort-3 proteins in all plants, trying to see what the differences are coming from, and trying to test specifically which proteins, which escort proteins may be responsible for this change in the budding profiles. And with that, I would like to thank our collaborators, Angela and Felix Frey in her lab. Tatiana Marquez and Andre Yers have been helping us with the diffusion and understanding the cargo retention capacity of these different budding modalities, and everyone in my lab. Thank you so much for your attention.